Okay. All right, thanks for coming. Today I'm going to be talking about sex chromosomes and sex determination, so different systems of sex determination. And before we start, I think I'll once again go over what we have. I'll probably do this every time from now on, just to keep us all on the same page. So just to remind you guys that next week is going to be the second quiz of this class. And it's going to be on everything since the last quiz until today. And it should be about the same format and length as last time. So I'm not going to make it too difficult. And then after the quiz, whatever I didn't get to today, assuming there's quite a lot of material on sex chromosomes, which we may not get through it all today. So after the quiz, I'll probably continue lecturing about sex chromosomes in the second part. Then after that, July the 4th, is going to be a class on different methods for studying chromosomes, and I think I'm going to emphasize what we're doing in my own lab for that. And I'll be passing out some 3D glasses. This is a simple kind with red and blue, so that we can see some three-dimensional images of chromosomes that we're taking with three-dimensional microscopy. That should be interesting. And then the last real class will be July 11th, where there will be kind of mini unit on the connection between chromosomes and cancer. And then there will be an overview, kind of like a study prep for the final test on July 25th. And we don't have any class on the 18th. Okay, so talking today about a very special topic in chromosome biology, and it's another topic with a pretty long history in chromosome biology, and that is how chromosomes affect sex determination and how inheritance is linked to sex. So first of all, just to get the terms right, so when we refer to biological sex, it's really the main, because there's different animals, there's different plants. You can't take male and female in humans as just the archetype of what biological sex means. It really refers to the size of gametes. That's the most universal aspect that's shared among all males and all females. So if you make a gamete that's this big, then you're a male. And proportionally, you make a gamete that's this big, then you're a female. And so there are other organisms like yeast and some single-cell organisms that have different mating types, but they're just single cells. They don't change their size. They don't have this asymmetry of gamete size, and so we don't refer to them as male or female. But fungus and plants and animals have this sort of independently evolved asymmetry of gamete size, so we say there's males and females. When you have males and females, and if you look at humans, we're a good example of it, and most other animals and plants have this ratio where the species is about half male and half female. And it may seem to us that that's obvious. Of course, we're half male and half female. But to population biologists in the middle of last century, that was actually a very interesting question. Why should that be the case? Why do we have 50% male, 50% female? And there's a pretty elegant mathematical argument developed by this scientist, Fisher, where if, for instance, if one sex were rare, if males were rarer, then it would be an advantage to produce more males. So if you produce only male offspring, since there aren't many males to go around, then the males would have an advantage in the next generation. So you would expect selection for genes that favor production of males. But that can't go too far, because then females would be rarer, and then you would select for genes that favor production of females. And so those two forces will just even out at a 50-50 ratio. There are a few more complications than that, but in general, if a male and female have about the same size and the same cost to raise from infancy to adulthood, then we can expect that the sex ratio in any species will be 50-50. And therefore, that ratio is not left to chance. That's something that's so important. We don't leave it to chance. There's actually mechanisms that try to maintain this sex ratio of 50-50. So it's not just random chance that there's half males and half females, but rather there are mechanisms that enforce that. And so also when we think of sex, we often think of sex chromosomes right away. We think, okay, XY is male and XX is female. But you guys probably know that there are many, many exceptions to that. And then there are a lot of animals that don't even use sex chromosomes at all. So a lot of reptiles, like these alligators, in fact, they lay eggs with no determined sex to begin with, and depending on the temperature at which that egg develops, it will either turn into a male or female. And in most cases, it goes kind of like this. If this is the temperature, then the extreme sides will be one or the other. Maybe the extremes of heat will be males, and the ones in the middle will be females. But the opposite arrangement happens as well. And so they actually have behavioral mechanisms where they'll monitor the temperature of their nests, and they'll arrange the eggs in it such that they have about 50-50 males and females. And that's pretty successful because alligators have been around for tens of millions of years and pretty much unchanged. But this is a little complicated, and it only works if you have this nest and you can control the temperature. So most mammals, in fact, I think all mammals, and most other animals use a different system. They don't use the temperature of development, but they use what's called chromosomal sex determination, which is what we're all familiar with, where depending on the chromosomes you have, you'll become either male or female. And usually it's either the presence of a sex chromosome or the absence of a sex chromosome, or in some cases, how many sex chromosomes you have. There are many, many variations on this. But depending on how many of these specialized chromosomes you have, you will determine your sex at a certain time in development. And we can speculate as to why. There are many ways you could potentially determine sex. So why so many times did chromosomes become the focus of sex determination? And some people think it's because this Mendelian inheritance, where you split alleles at meiosis, gives you a very easy way of achieving a 50-50 ratio. You either inherit something or you don't. That's automatically a 50-50 ratio. So you don't need to do any tricks like monitoring the temperature or relying on some other way to generate a nice 50-50 split. You get that automatically with Mendelian inheritance. And I should say that although we call it chromosomal sex determination, it's not that the entire chromosome has this maleness or femaleness to it, but rather the sex chromosome will have a certain number of genes on it, and it's inheriting those genes that lead to a particular developmental outcome. So we have sex chromosomes, and other reptiles also have sex chromosomes, but alligators don't. Birds do, and birds are very closely related to alligators. But so where did they come from? How did they get their sex chromosomes? And a long time ago, 100 years ago, the suggestion, this was just a theoretical suggestion, was that, of course, they had to have come from the autosomes, from the non-sex chromosomes that were there to begin with. And the idea is that once you have, so let's say it would just be a typical chromosome, like chromosome 2, for instance, but one of the two chromosomes would have a mutation on it. Let's call it M mutation, which will lead, let's say it's a dominant mutation that gives rise to males. So if you have this mutation, then number one, you don't want this mutation to spread throughout the entire population, because an entire population of males is no good. So you would tend to have some system where you only want one copy of this chromosome, because having two copies in the entire population would wipe out the population. And additionally, if you start to use this allele to determine the male sex, then there will be strong selection for other genes that have to do with male production, such as genes that code for sperm proteins or or male hormones or things like this. Those sorts of genes, if they happen to land next to this male determining gene, they will have an advantage because they'll always be present in the male. So the idea is that you get linkage to a big, not just one gene that determines males, but to several genes that are only good if they travel together into the male body. And so if then another event has to happen, which is you have to somehow suppress recombination. So this part of chromosome 2, for instance, like we talked about last time, you might get some sort of inversion where the entire thing flips around. Then it will not be able to recombine with the other chromosome 2, so it will be left as a kind of genetic isolate. And this part of the chromosome then becomes, even though it's still attached to the rest of chromosome 2, this segment of the chromosome would start to just be like one independent gene, one big super gene. And eventually, this region of recombination suppression can grow and grow, and then eventually this chromosome that started off just having one gene on it, eventually instead of being chromosome 2, this now becomes a Y chromosome. So once it's changed enough, once this part that determines maleness has gone through enough generations and accumulated enough changes, then it no longer behaves like chromosome 2, but it's you can consider it now the Y chromosome. And the original chromosome 2 now, you can call it the X chromosome. And yeah, kind of this is sort of the schematic for that I was through there. But right, it only takes one gene, theoretically, that if this gene here, this gene, it says TDF, which would be a testis determining factor. So it's a gene that results 100% of the time in producing testis. So that's what makes them a male. If you have that one factor, then many other genes will start to accumulate next to it. So how do the other genes accumulate? Well, just over evolutionary time, chromosomes can break and refuse. So genes are not always located on one chromosome, but very rarely they can jump from one chromosome to another. And if they just happen to jump next to that testis determining factor, then there will be an advantage in the next generation. Okay, so the idea is that you start off like an alligator, where you do not have a chromosomal sex determining system. But if you happen to get one mutation or one new allele that leads to maleness 100% of the time, then the rest of this naturally follows. And that chromosome that got that mutation first will become the Y chromosome, and the other one becomes the X chromosome. So that system, and we know that this has happened because we can look at these other parts, and I'll show you more in a minute. But the entire chromosome doesn't go away. There's bits of the chromosome left that still resemble the original chromosome that it was derived from. And these are actually very important, that you maintain a little bit of identity, that you do not completely destroy the original chromosome, but there's some bit that's remaining. So we know that this came originally from this chromosome. So that's a process that's the process that creates sex chromosomes. But sex is only one trait of the many thousands of millions of traits that animals have. So why is it only sex chromosomes or sex determination? Why is that the only system that can evolve like this? Why doesn't height or some other phenotype get its own chromosome as well? So first of all, one answer is that it's not, right? There are other chromosomes. There are other super genes like this. And one case was described just 1 year ago. There's a bird called a ruff that those three illustrations are actually 3 different kinds of male bird. So it's not male versus female, but there's 3 different kinds of male that have different kinds of feather patterns. The one on the right actually looks almost identical to a female, but it is a male. And it's not only the way they look, but also the way they behave, and their mating strategies are all controlled by this kind of super gene, which is like this, but it's not degenerated quite as much. But if I don't know which chromosome it's on, but if this is the normal chromosome, then there's one region that's flipped. So part of this chromosome is flipped around, and the genes that control all this behavior and feathers and everything that determine the difference between those males are all in this super gene locus. And given enough time, and if the distinction between those 3 kinds of male is important enough, then over time this might become its own chromosome as well. And so it would not be a sex chromosome, but it would be a mating behavior chromosome. But however, that is still a sex-related phenomenon, right? This only happens in males, and this feather and behavior complex really is all about how they mate. It doesn't affect any other part of their life. So sex does seem to have this kind of driving factor that it tends to do strange things to the chromosomes. I won't get into details, partially because I don't really understand all the details myself, but this phenomenon of meiotic drive, where you can have a non-Mendelian effect, where one allele will preferentially go into the next generation, it seems that sex determination mechanisms are particularly strong in this meiotic drive. So probably because, well, I'm not really sure why that is. Probably just because, probably because it has to do with which kind of gamete you make. So, yeah, I don't know. This meiotic drive happens in oocytes. So, yeah, well, I don't really know. I thought I had the answer to this, but somehow sex determination and meiotic drive are linked. But I'll have to look at that mechanism next time, because actually that's something I forgot to research here. But yeah, okay, yeah, I'll have to look that up for next time. Sorry, no, that's good. But okay, so anyway, let me just. The point here is that it's not just sex determination that can give rise to this chromosome degeneration phenotype, but other phenotypes can cause the same sort of super gene cluster that a sex chromosome is one example of. And why sex determination does this more strongly has something to do with meiotic drive, but I don't know what it is. I'll have to figure that out for next time. Okay, so when we talk about chromosome sex determination, a phenomenon that comes up a lot is sex linkage, or traits that are carried on a sex chromosome. And here we're kind of getting into the evidence for the existence of sex chromosomes and the linkage between figuring out that sex chromosomes were a thing, that they existed, and the chromosome theory. So this section is from kind of 100 years ago, where people were still working out that chromosomes were the genetic material and that they were the units of heredity. And sex determination and sex linkage played a big role in figuring out chromosome theory. So when we think about the connection between phenotype, between the animals' bodies and their sex, there's 3 different phenomena that can be associated with that. The first 2 are kind of similar. So sex-limited versus sex-influenced traits just a sex-limited trait would be something that only ever appears in males or only in females. So some animals might have horns only in the male, but not in the female. So you might think, okay, this is an example. It's only showing up in males, so does that mean it's on the sex chromosome? Does that mean the horns are coded on the Y chromosome? Well, no, that does not mean that. It's just the genes are present. It's probably autosomal genes that determine the presence of male horns versus the lack of horns in females. But there's some developmental rule that happens as the embryo is developing, where they get a signal, and if those genes are in a male embryo, then they'll make the horns, and if they're in a female embryo, then they won't. Maybe they'll make something else. So this is something where it's only ever in one sex and not the other. And then similarly, there's a sex influence trait, which just means that the degree of expression is different in males or females. And you might think this too might be on the sex chromosome, but actually, similar to number one, there's just a developmental rule that decides, okay, females are going to modify this trait in a certain way, and males are going to modify it in another. So a male lion will have this big mane. Well, a female still has hair, but it's not developed in the same way. Okay, but then finally, there's sex-linked traits, where these are more simple traits. They're usually one or two genes that determine this entire trait, and it itself is linked to the same system that determines sex. So here there's no developmental rule because it's just the gene is either there or it isn't, or it's either mutated in the case of recessive genes, or it isn't. The way the sex-linked traits were first kind of figured out was by their different behavior when you cross a male versus female than when you cross a female to a male. So what I mean by that is, let's say you have a mutation, and you don't need to look at all the details here, but this was first figured out in Drosophila, where there was a mutation where the Drosophila, these fruit flies normally have red eyes, but there was a mutation where the eyes were white. So they don't make this red pigment in their eyes, and it was a clear mutation. And if you kept mating these flies, they would always have white eyes. So if you look at the section on the left, if you cross a wild-type red-eyed female to a white-eyed male, then in the next generation, so 2 generations down, if you look at all the progeny, only the males, only white-eyed flies are male. So there are no red-eyed females in the 3rd generation. So you start with these parents, you have equal numbers of red females and white males. You mate them, but then 2 generations later, there are no white-eyed females, there are only white-eyed males. But if in the 1st generation, so over here, if instead of crossing red-eyed females to white-eyed males, you do it the other way around, white to red, then 2 generations later, you have both white-eyed females and white-eyed males. So if these genes were on any other chromosome, or if it were just a developmental switch, if it was, okay, if you're a female, then turn on red eyes, and if you're male, then turn on white eyes, then it shouldn't matter which way you did the cross in the 1st generation, because if it's just based on your sex changed eye color, then there's no way to know what was happening 2 generations ago for these guys. But if you assume that chromosomes carry something, and males have only one of these chromosomes and females have 2 of them, so if you just assume this sort of theoretical construct, then that explains it. So meaning that if males only have 1 X chromosome and females have 2 X chromosomes, and that chromosome is where this eye color is located, then that explains this inheritance pattern. And so similarly, a lot of human pedigrees can be figured out in the same way. So we happen to have the same kind of sex determination system as flies, so males have X Y and females have XX. So for instance, if a male has some mutation and it only shows up in the man's daughters, but it does not show up in the man's sons, then you can assume, okay, it has to be an X linked, it has to be on its X chromosome, because the sons, of course, do not get the father's X chromosome, but all his daughters do. Whereas, if a woman happens to have this recessive mutation, and if only her sons get it, so if a man can give something only to his daughters, or a woman can give it only to her sons, in either case, that can only be explained by this chromosomal linkage. So this mutation is located on the same chromosome that is directly responsible for determining the sex of the offspring. Okay, so it's not just humans and flies, but most mammals in general use this XY system. The particular mammalian version of this XY system is what actually determines its sex is whether or not you have a Y chromosome. So the amount of X chromosomes do not seem to matter. The typical chromosome configuration is XY, so most males are just XY. But if you have an extra X chromosome, that doesn't change anything. You're still male. If you have 2 extra X chromosomes, even there are some differences, sure, but you're still mainly biologically male, and you'll still have a gonad that produces sperm and not eggs. As far as we know, I think most mammals, I wouldn't say 100% because there's always exceptions, but most mammals work out this way. We talked about this a little bit before in the sense that we have some flexibility when it comes to the number of sex chromosomes that we can accommodate. So having extra X chromosomes, for instance, you're still a male, but there are some health problems associated with that. So XXY is a male with Klinefelter syndrome. If you don't have, so also females, you don't actually need 2 X chromosomes. We usually have 2, but you don't actually need 2. If you only have 1, then we have what's known as Turner syndrome, and that also leads to some health problems. And actually having extra Ys does not actually seem to give any problems. People with an extra Y chromosome usually don't even know that they have it. The same is true with females with 3 X chromosomes. Okay, so it's really just the presence of a Y will give rise to males and humans and most other mammals. And just an aside, these XY chromosomes, although some inheritance patterns have been seen before, there's this American cell biologist named Nettie Stevens who actually discovered XY chromosomes just by looking at them. She didn't discover them in humans, but actually in these mealworm beetles. And that's a picture of the microscope she actually used to make this discovery. And it was last year that I just randomly opened up Google, and it was maybe one of her anniversaries, and so they had an illustration of her showing some Y. Okay, yeah, so that was about 100 years ago that these were found. Earlier I said that it's not the entire chromosome, it's not the entire Y chromosome that determines that someone is going to be a male. But there's actually a single gene, just like that single testis determining factor, that is still present on the human Y chromosome and mammalian Y chromosomes that encodes this special protein called a transcription factor, and that determines the sex. And this transcription factor called SRY is a protein that goes to the chromosome and binds to the start site of many, many other genes and turns them on. And it's the kind of protein you would expect to be a master regulator of something like sex, because it turns on all these other genes that are required for male formation. And those genes are not only on the Y chromosome, in fact, they're mostly not on the Y chromosome, they're on all of our chromosomes. The way this was found was by just taking just this SRY gene, just this little part of the Y chromosome, and putting them into mice that have 2 X chromosomes. So there's no need for the whole, the whole Y chromosome, but just this little part, this SRY, is sufficient to turn on the male developmental program and turn these XX mice into males. Unfortunately, those mice are sterile because there are some genes on the Y chromosome, some of those linked genes that are needed to make sperm completely. So other genes on the Y chromosome are needed for full fertility, but just for turning on the male developmental program, only that one gene, SRY, is sufficient. Okay, so what does that look like? So in the case of humans, this is kind of a global zoomed out view of our sex chromosomes. So there's that on the right there, the Y chromosome has that SRY location. And the Y chromosome isn't actually that much. I mean, it's one of the smaller chromosomes, but it's still bigger than some of our other chromosomes, so I'm not drawing it too small. So there are some regions on X and Y. I've drawn the centromeres because they both have centromeres. They also have telomeres, of course, but they also have these regions called PAR1 and PAR2, and those are also quite important for X and Y chromosomes. So remember I said that these, even though this chromosome degenerates and loses pieces of itself, it always has to maintain some regions that are still the same as the chromosome that they derived from. And there are some genes in here as well. And so if you think about that earlier picture I showed of human, this human pedigree, for instance, if one of the genes, if there's a gene in here, then even though it's on the X chromosome, it's not going to show this kind of skewed inheritance. It's going to look like a normal autosomal gene. It'll look like any gene that lives in here is going to behave just the same way as any other dominant or recessive gene on an autosome. And that's why these regions are called the pseudo-autosomal regions, because any gene that you have there, you would not know it's on a sex chromosome unless you actually sequenced it and found its location in the genome. So that's kind of what was observed, but what are these for? Does anybody think why it's important to maintain these regions? Yeah?
Is it so they can match up during sinuses?
Yeah, that's probably the main and maybe even the only reason. So so the Y chromosome over time, since it's not recombining with the X, it's changing, going over many generations and losing almost all the information. But you're right, at meiosis, of course, chromosome 1 can find chromosome 1 because it's homologous. Chromosome 2 can pair with chromosome 2. But what happens to the X and the Y? Right? The X and the Y are different chromosomes. So do they pair with each other? Or do they somehow manage to do meiosis without pairing? Well, yeah, actually they use these pseudo-autosomal regions, pairing at one or both of them, to do some recombination. So they do still do recombination in here, and that's how they're maintained over time, is because they keep swapping pieces. But more importantly, it makes that connection, it makes the chiasma that allows the X and Y to point in opposite directions at meiosis, so that then they can go to opposite daughter cells. So that's where you get the 50-50 ratio, because the X goes one way, the Y goes the other way, right? So, hang on. I mean, that might be why meiotic drive works with these guys then, because you have different sizes of chromosomes, and you have the oocyte with different sizes of egg and polar body. I bet that's probably the reason. I will still check to make sure, but now that I think about it, that's probably the reason why once you have unequal sized chromosomes, you can have some meiotic drive happen. Okay, so yeah, this is kind of a basic overview of human X and Y chromosomes, and it works the same way. So all mammal chromosomes have these pseudo-autosomal regions that undergo meiotic pairing, and they also have normal genes that, maybe for all they knew, they don't even know they're on sex chromosomes. They just They just behave like normal autosomal genes. So in contrast, so Drosophila also has X and Y chromosomes. So males are XX, and so they're that a fancy word for that is heterogametic, which means they have two different kinds of gametes. They have sperm with an X and sperm with a Y, so they're heterogametic. And females are XX. But here there's just a slight twist, and that is that it's not actually the presence of the X or Y of the Y chromosome that makes males, but rather whether there's one or two X chromosomes. So if you're an embryo and you start to develop, if you're XY, you will turn into a male. If you're XX, you turn into a female. But yeah, I think X0, I didn't list it there, but XO is also a female, so it's not going to turn into a male. Sorry, XO is also a male, right? Because it's only one X chromosome. Sorry, yeah, X and no Y. So without the Y chromosome, it's still turning into a male. Okay, right. So it's not that the Y chromosome carries this special male determining factor, but rather there's some mechanism that can count whether there's one X chromosome or two X chromosomes. And I'll talk more about chromosome counting systems later. So that's just a little wrinkle there. And there are other mechanisms of sex determination in other insects. So not all insects, not all animals, and definitely not even all insects work that way. For instance, there's these eusocial hymenopterans, bees and wasps and ants, that have this what's called a haplodiploid sex determination system. So here there's not even a special sex chromosome, but it's rather how many sets of chromosomes you have. So last time I talked about, so we've gone over some terms like haploid and diploid. So this is the number of chromosomes in a gamete, and this is the number of chromosomes in the zygote. So in this system, it actually there are grown adult organisms that are either haploid or diploid. Normally, it's only our gametes that are haploid. Only sperm and eggs are haploid in most animals. But here we have a case where the adult animal is still haploid. It still only has one set of chromosomes. And how does that work? As you can see that, so females, of course, they lay eggs. Some of those eggs are fertilized, and the fertilized eggs develop into other females. Whereas they somehow keep other eggs that prevent them from getting fertilized internally, and they lay a certain number of those eggs that naturally turn into males. So again, the embryo, as it's growing, it has to have some mechanism to know how many sets of chromosomes they have. Do they have two sets of chromosomes or just one? I'm not really sure what that mechanism is. So basically, as you can see, this male comes in on the right there, and if he gives his A, B, C, D chromosomes to this female on the left, then none of those chromosomes will make it into the next generation of males. So only the original chromosomes of the female are going to come into this next generation. So that's just another completely different system. Then there are other animals, and you probably are aware, some insects use a similar system like this, but also most birds use this system. It's actually kind of the mirror image of the XY system. It's only called ZW just to distinguish it from the X and Y system. We could just as easily call these X and Y chromosomes. There's nothing special about their chromosomes. It's just, in this case, males are the homogametic sex. So males only make one kind of sperm. Those are the sperm with Z chromosomes. Whereas females make two different kinds of eggs, some with Z and some with W. And here it's kind of like the mirror image of mammals. So the presence of the W chromosome determines whether the bird grows up to be male or female. And so birds are pretty relatively closely related to mammals. If you go back long enough, of course, we're all just this. There's some ancestral amniote, some organism that developed from an egg. And right, so if you look, so doing this kind of fluorescence in situ hybridization, where you can take individual chromosomes from birds and say what regions in mammalian chromosomes do those map to, and vice versa. You can take individual sex chromosomes from mammals and say where do those map to in birds. You can see that they do overlap quite a bit, but they do not overlap. They don't overlap with the sex chromosomes. So in other words, the bird sex chromosomes, Z and W, if you make a fluorescent probe that will highlight this, or if you just do genome sequence and look where the sequences map, it's kind of the same thing. You will see that the bird Z and W map to different autosomes of different mammals. This one on the left here is an Australian marsupial, pretty far back. It's pretty distant relative of humans. And this one is humans, and I suppose mouse would be the same. So we do have chromosomes that are related to these bird sex chromosomes, but as you can see, they're autosomes in us, which means that it was thought that the original evolution of sex chromosomes, sorry, I don't have a pointer, but it had to have happened at some point after that 310 million years ago. So somewhere it happened once in birds on that side, on that branch, and then it happened again in mammals on that branch. Because also if you take the mammalian X and Y chromosomes, or the wallaby X and Y chromosomes, there are homologs, there are partners, they match some chromosomes in birds, but they don't match the sex chromosomes, they match parts of autosomes. So it's thought they were completely independent until people did the same sort of genomic mapping on another interesting organism called the platypus. So Optulce. So Optulce has very, very strange sex chromosomes, but in fact, its sex chromosomes, you can see multiple X's and multiple Y's, they almost exclusively have the bird sex chromosome material in them. And if that's true, if their chromosomes are sort of related to the bird chromosomes, then there would have to have been some common ancestor, maybe earlier than this 310 million years ago, that originally had this sex determination system. And then along the line somewhere to mammals, it got fragmented up into all these pieces, and then there was a switch where these sex chromosomes stopped being sex chromosomes altogether, and instead these autosomes then newly turned into sex chromosomes. So it's still not really clear which is which, but it's worth looking at actually this platypus itself as to how this thing actually works, because so it has 5 different sex chromosomes. It has 5 X chromosomes and 5 Y chromosomes. And it determines sex in the same way as mammals. So if you have the Y chromosomes, you're a male. But the crazy thing is that in meiosis, when they undergo pairing, they actually do, the chromosomes all link up together in this chain. And they pair together in the same order all the time. As if originally there was one big chromosome that fragmented into pieces, but still at meiosis, it knows how to reassemble into one giant mega chromosome. Because at the end, all 5 X chromosomes, or all 5 Y chromosomes, are able to go to daughter cells. So they make, they link up like this, and they actually somehow manage to go into opposite cells. So they don't, somehow they maintain this balance of X and Y. So platypus is really just weird. And it's just an example. I've kind of given up trying to understand how it evolved. But the fact is that sex chromosomes, probably because of this meiotic drive phenomenon, they really can evolve quickly. So even closely related organisms can have very different sex determination systems. So you really have to kind of keep an eye on. You can't assume, if you were just to discover a new species, new mammal species, you wouldn't necessarily assume that sex determination worked the same way. And then, yeah, so we've seen kind of an example of this before with this praying mantis. Remember I showed you that there's 2 X chromosomes and 1 Y chromosome in this guy, and this was used as an advantage because sometimes these 2 X chromosomes don't come together, and you can pull on one of them to make meiosis happen. So this sort of platypus-like thing with multiple sex chromosomes, it's not unique to the platypus. It's something that happens in many lineages. So the point is we can't really think of our XY system as normal and other systems as the freak systems. In actuality, they're probably constantly undergoing change and evolution, and they're probably not stable. So our system may change at some point in the future as well. Okay, and then the last system that I'm going to talk about, which seems at first like a simple system, but actually we know a lot of the molecular details about this, so I'll talk about that, is the XO system. So this system actually does not involve the Y chromosome at all. It's kind of like the haplodiploidy system with insects, where it's the number of chromosome sets that determine the sex. But in this case, the counting is a little bit more simple. So it's called the XO system, but the O just really stands for nothing. So animals have either a single X chromosome or 2 X chromosomes, and those are the 2 possibilities. And that is the determining factor, in fact. So animals with a single X chromosome end up males, and animals with 2 X chromosomes end up as females, or in the case of many nematodes, many worms that have this system, they're not actually females, but they're hermaphrodites. But we can think of them as females. Sometimes they're females, sometimes they're hermaphrodites. And this is the system, in fact, that C. elegans, the worm that I've introduced before, this is the system that they use. So it's all about the number of sex chromosomes. But at first it was thought, okay, it's simple. If you have 2 X's, then you become hermaphrodite, or else I'll say female. And if you have 1 X, you're a male. But after people investigated the mechanism for how this happened, it turned out that it's really not so simple. It's not just the number of sex chromosomes. In other words, there's not something that's counting, and it says if you have 1, you're a male. Rather, what's measured is the ratio of X chromosomes to the number of sets of autosomes. In other words, if we look at this constitution, so this is a typical C. elegans male. It has a single X chromosome, and then it has 5 pairs of autosomes. In other words, there's 2 complete sets of autosomes. So we can say that the ratio of X to A, we call this the X to A ratio, is 1 to 2 in the case of males, and 2 to 2 or 1 to 1 in the case of hermaphrodites. So why is it the ratio, or how do we know that it's the ratio of X to autosomes and not just the number of X chromosomes? And of course, it happens from looking at if this were the only cases that people looked at, then there would be no difference between number of sex chromosomes and the ratio. But there are other numbers of chromosomes that we can look at. For instance, so I've already talked about haploid and diploid. but there's also the potential of being tetraploid, right? So haploid, and then tetraploid. So haploid means you have one N. Diploid is 2N. And tetraploid is 4N. Okay. So yeah, tetra just means 4. And you can be higher, you can be hexaploid, octaploid, decaploid, dodecaploid. It's always a Greek number that you put in front of ploid. And so anyway, if you use a certain drug, you can stop the first meiotic division as it's happening. It's actually a drug that attacks microtubules, so it dissolves this entire spindle. So as the cell is dividing, instead of dividing, it all just falls apart and the nucleus comes back together. The cell thinks it's divided, but actually it still has all 6 pairs of chromosomes. It has all 12 chromosomes. Then it undergoes the second division normally, and it results in a gamete with 12 chromosomes instead of 6 chromosomes. And if you start with that gamete, and then you have an egg like that, a sperm like that, you put them together, and now you have a worm with 24 chromosomes instead of 12. So you pretty much canceled out meiosis and just doubled the genomes by mating 2 worms together. So in this case, the ratio, in the first case, that just doesn't change anything, right? The ratios are the same. So now the hermaphrodites or females will have 4 X chromosomes and 4 sets of autosomes. We've just doubled everything. Males will have, this should be reversed, sorry, 2X, 4A. Yeah, that's wrong. So the ratio in males is still 0.5. The ratio in females is still 1. So we haven't learned anything yet, but now the trick is we'll finally start doing some interesting things when we make diploid times tetraploid and end up with triploid. Because now we can start to get different ratios of X chromosomes to autosomes. And so when that was done, so some of the ratios are still the same. Okay, if you're just a straight up triploid hermaphrodite, you have 3 X chromosomes and 3 sets of autosomes, then you're still fine. You're still normal hermaphrodite. And the other cases we've covered already. But now there's 2 more interesting cases. One is if you have 3 X chromosomes and 4 sets of autosomes. Now we have a different ratio. It's 0.75. And these guys happen to develop also as hermaphrodites. So if we keep that ratio, then these guys develop as hermaphrodites. So clearly we can have, so it's not just, right, so there's 3 X chromosomes, 4 sets of autosomes. They're different, but we're still hermaphrodite. However, in this case, so this is a triploid worm, but except it only has 2 X chromosomes. There the ratio is 0.66, and those always develop as males. Okay, so what we have on this side is everything here is developing as a hermaphrodite. Most of these ratios are 1, but it can dip as low as 0.75 and remain hermaphrodite. On the other side, these 2 ratios are both 1/2, 50-50, but it can go up as high as 0.66 and stay a male. So somewhere along that dividing line, if the ratio of X chromosomes to autosomes is 0.75 or above, then that will always become a hermaphrodite. If it's 0.66 or below, then it's always a male. So when people figured that out, then the next natural step was to explore a little further and try to get ratios in between 0.75 and 0.66. They could have done that by trying to make more sets of autosomes, so trying to maybe go one level higher, make a hexaploid worm or something like that. But it turns out worms don't really like to have too many tetraploid is about as high as you can go with worms. If you try to do that trick again where you disrupt the spindle and try to double the number of chromosomes again, you will just have too many chromosomes and the worm cannot survive that way. So, in other words, to look closer at this ratio, instead of changing the ratio by a whole chromosome or a whole set at a time, what was done was small parts or small chunks of the X chromosome were added in separately. And how's that done? So these, there's a really big resource, a genetic resource of this nematode, C. elegans. People just bombarded these worms with gamma rays and broke up all their chromosomes and just cataloged, okay, we have a piece of the X chromosome, this extra piece of the X chromosome floating around, and it was found by a certain lab that's identified by the initials MN, and they made DP there stands for duplication. So they made new worm strains with these extra little duplications of the X chromosome. And here it's really an advantage that C. elegans does not have centromeres, because these duplications can just get transmitted from one generation to the next. And you can have duplication from anywhere in the chromosome. You don't need a centromere for them to be transmitted. So anyway, yeah, they had a whole series of duplications, starting from 1, and I think that probably goes up to 50 or so. And so they decided, instead of adding an entire X chromosome, they'll add these fragments of the X chromosome and see if those affect this sex determination ratio. So the ones I'm going to discuss are MN-Dup9 and MN-Dup10. So first of all, starting with this guy, so he's right on the edge, these 2X, 3A, so the ratio is 0.66, and they always turn into males. But if you add this small bit of the X chromosome to those males, then the offspring are really right on the cusp. So they are about half hermaphrodite and half male. So adding this little bit of the X chromosome just tweaks that ratio a little bit higher, and now there start to be some hermaphrodites in the progeny. And if you add this one instead, if you add this larger one, then it becomes 100% hermaphrodite. So in other words, adding this piece of the X chromosome will lift this ratio up a little bit more, so that it goes from all male to all hermaphrodites. Right. However, yeah. So you might think, okay, does this have as much hermaphrodite generating power as an entire X chromosome? Because of course, if you add an entire X chromosome to this, if you make it 3X, 3A, then of course there will also all be hermaphrodites. So does this contain all the sequence or all the genes needed to count as a full X chromosome? Well, it actually doesn't, because if you take a normal male that only has one X chromosome and 2 sets of autosomes, and you add this to it, then that's not enough to produce hermaphrodites. In other words, so this is enough to push this 2/3 guy up to 100% hermaphrodite formation, but it's not enough to push a 0.5. So it's a little complicated, but so if you're 0.5 plus MN-Dup10, that's still all males. But if you're 0.66 plus MN-Dup10, then that's all hermaphrodite. Whereas if you add an entire X chromosome to either of these, then it will be all hermaphrodite. So in other words, this small duplication has a little bit of hermaphrodite generating power. This bigger duplication has a little bit more power to generate hermaphrodites, and the entire chromosome has the most. So it's as if there's this graded sequence where the more X chromosome you have, in these edge cases, the more likely you are to generate hermaphrodites. So the next question is it, so for counting this ratio, is it actually just the amount of DNA that you have? So is it really like X chromosome DNA is counting towards this ratio, and it's just how much of it do you have? Or another model is that it could be actual genes on these chromosomes, and you need a certain number of genes, right? So those are two models. Is it just how much of the chromosome you have, or are there actually certain genes where this chromosome has one gene, so that gets you part of the way. This chromosome has two genes, that gets you a little bit more, whereas the full X chromosome has three genes, and that gets you all the way. And in fact, the evidence turned out that it is actually this discrete model, and it's not just, well, how much chromosome do you have, but rather there are these discrete single locus elements on these chromosome duplications that count toward this ratio. And the way this was found was by, of course, looking at other chromosome duplications. So looking at chromosomes that are a little bit longer than MN-dup10, but not as long as the full X. This lab made really many of these duplications, and they know how big they are. So if you add a little bit extra, that doesn't change the hermaphrodite generating power. You have to add as much as this. You have to, once you get to a certain critical length, then that will boost up the level of hermaphrodites by one more level. Likewise with this one, this growing this duplication by a little bit did not change the number of hermaphrodites, but once it achieved a certain threshold, then the ratios changed. So in other words, there were these threshold lengths that were identified that these markers were genetically mapped that way by how much chromosome you needed in order to see a visible difference in the number of males and hermaphrodites. So really, these experiments were done before too much microscopy was used and before genomes were sequenced, and they're really just people counting numbers of males and hermaphrodites based on what kind of X chromosomes they had. But this was really figured out so that there were actual, this led to genetic evidence that there are these genes on the X chromosome, and it's actually the number of these genes that makes up this ratio, the number of X response elements that act to determine the sex. So the X chromosome has a certain number of genes on it, and the autosomes all have also a certain number of genes on them that act to So just a little bit go into the detail about what these elements do. So each of these X response elements in turn. So you might think, okay, there are different genes, so they all have different functions, and if they have different functions, then how can they all sort of work towards the same goal of making a male versus a hermaphrodite? So it turns out that even though there's different genes, these positions all encode for different genes. The main effect of those genes, they're all concentrated on turning on or turning off this one single gene called ZOL1. So there's ZOL1 was found out to be this kind of like the SRY gene in mammals. It's the master gene that leads to male versus hermaphrodite sex determination. So ZOL1 is required to make males. So these genes on X, they all act to turn off that gene. But they all act at a different part of that pathway. So in order to turn on a gene, you need several steps, right? So here's the chromosome. here's the gene ZOL1, and this is the DNA that's upstream of ZOL1. So in order to turn on this gene, so in order to make a male, you first need to have a protein binding to this part of the gene upstream. So this is a transcription factor that turns on this gene. Then you need to actually get mRNA made. So this gene has to get transcribed. Then, so that's step number 2. Then this mRNA has to get transported outside the nucleus. And then the mRNA gets translated into a protein. That's step number 3. Then this protein has to survive long enough to come back into the nucleus. And then once it makes it into the nucleus, then it will, like SRY, it also acts as a transcription factor and turns on other genes that will eventually lead to male production. But it turns out that each of these steps has a gene that can come in and disrupt that process. So there's another protein that comes in and actually disrupts this transcription factor from binding. There's another gene that specifically comes in to block transcription of ZOL1. There's another gene that prevents this translation. There's another gene that wants to come up and chop up ZOL1 protein. So ZOL1 is a typical gene, but somewhere on the path between this gene and making it back in to turn on the male genes, it's under constant attack from all these other genes in the worm. And these genes that attack ZOL1 are these X response elements here. Okay, so just the more of these genes you have, the greater the chance is that you're going to suppress ZOL1 somewhere along the way and stop male development. Whereas if you don't have enough, if you don't have too many of them, then there's enough ZOL1 escaping, and if you have more autosomal targets as well, so the more autosomes you have, then the greater chance is that ZOL1 will get in and turn on the male genes that are required. So the autosomes act to enhance male production, whereas the X chromosomes act to break this assembly line of ZOL1 creation. And just, each step, of course, is the result of years of genetic experiments to show that this is happening. But yeah, this is kind of the nitty-gritty molecular biology of sex determination. So it's not just, okay, the X chromosome makes hermaphrodites, and if you miss the X chromosome, then you become a male. But the X chromosome contains these very specific genes that attack very specific steps along the pathway of creating this protein that will turn on male production. So yeah, it's actually a lot more complicated. It seems simpler because it's just X or no X, or 1X or 2X's. But having this counting mechanism actually makes it a lot more complicated than the mammalian system. Our mammalian system is very simple in comparison. We either have this SRY gene or we don't, and there's no need to turn it off because females simply don't have it and males do have it. Whereas worms, the ZOL1 gene itself is not on the X chromosome; it's on an autosome. So in other words, in humans, the SRY gene is a sex-linked gene, whereas in worms, ZOL1 is just a sex-limited gene. And that makes a much more complicated system in regulating sex determination in worms. Okay, so yeah, I think this is pretty dense. The next, so I've talked about almost everything in terms of sex determination. So I've said that the sex determination systems can change rapidly over evolutionary time, but it's not quite as simple as that, because changing the sex determination system to a chromosomal system will actually lead to many more problems, and it leads to a particular problem here at the very end. So once you get this chromosomal sex determination system, that will lead naturally to changes in sex chromosome, and in particular, the heterogametic chromosome, so the Y in mammals or the W in birds, starts to degenerate and lose information. When you get that, then you get this situation of heterokaryotypy, where males and females have different numbers of chromosomes. So that necessitates another mechanism. So those males have XY, females have XX. So for many, many genes, females have twice the number of genes twice the number of copies of those genes as males do. And yet, males and females are mostly identical at the cellular and gross physiological level. We're pretty much the same. So, in other words, this difference in gene dosage has to be compensated for somehow. So, it should be okay to have 2 copies of many of these genes if you're a female, and only 1 copy if you're a male. Some genes it's okay. It's okay for that dosage to vary. But other genes, it's could be very sensitive to that. So it's just like having a recessive mutation. If you only have 1 versus 2 working copies of a gene, that could actually lead to some problems. So there's this mechanism called dosage compensation that acts to equalize the gene expression levels. And it's also a pretty complicated topic, so I think I'm going to mostly talk about it next time. But just like sex determination mechanisms undergo rapid evolution, the mechanisms of compensating for different dosage also undergo a lot of changes. And the 2 things have to change together. But yeah, I think I can't really get into it and finish it in time today, so I think I'll just save it mostly for next time. So maybe it's a bit early, but this was a pretty dense topic. So if there's any questions, you can ask now or after I turn it off. So so as far as the quiz goes, all the details are not so important. I'm not asking you to memorize the names of these duplications or anything, but just the general gist of there are different sex determination systems, and birds use a different one from mammals, and crocodiles and alligators use a different one. I think if you have any questions about today or earlier lectures, you can feel free to ask me, of course. But if not, then we'll just end here. Yeah.
Why in Chrysopula is XXY?
Where is it? Yeah, I put that up there, didn't I? That's a good question. I don't know why that is. I think I'll have to check on that one as well. I guess, probably there are some genes on the Y chromosome that only go with male development, but it's the number of X chromosomes that determine the sex. So XY is okay because a single X makes you a male, and then you get these Y genes that are useful in a male. If you're XX, then you have to be a female, but then you have these extra Y genes that I guess somehow interfere with development. So it seems like it's kind of a bad system, right? That should be. But I mean, normally your XXY is a very rare thing. It only happens during this non-disjunction. So I don't know the actual molecular details of like which genes on the Y will kill females but not males, but there must just be some developmental pathways that are turned on that only work if you're a male, but they will leave them. With my PhD, the university where I got my PhD, the person who figured out this Drosophilus sex determination system and the person who figured out the C. elegans sex determination system were both in my department, and they're actually married to each other. So, but I was a worm person, so I never really got into the details of the fly system. But they're both really complicated, but and both very different as well. But yeah, the fly system, their gene is not ZOL1, but it's a gene called sex lethal, and it has kind of the same thing. So I think it may be a gene on the Y that causes death in female flies. Yeah, a little early today, but I don't mind. You can be dismissed if you don't have any questions.